Hi, everybody. My name is Rafael, and I'm here to talk about what we've built over the past months. The title of my presentation, actually, I stole it a little bit from In Silico, where they have their headline called From Prompt to Drug, but this is From Prompt to Peptide. Agentic, sorry? It's much better, the PNP. So anyways, agentic science for peptide design and discovery. The question that we ask ourselves is basically what changes when design, validation, and the wet lab stop being separate stages and start running as one integrated loop. The inspiration was basically that we saw that autonomous science is already delivering. In February, we saw the paper that came out of OpenAI working with Ginkgo Bioworks, where basically they increased cell protein synthesis yield and also the time that it would take, the cost that it would take in about 5 months from scratch. They gave access to their automated lab to GPT-5, and in 6 months it delivered a paper. More recently, 2 days ago, a company also called Molecule, but Molecule.1, that works on automated chemistry, gave access to GPT 5.4, and it increased the yield of very hard chemical synthesis reaction for 88%. So it's already happening. But why now? There's 3 things mainly. There's more, of course, that are true at the same time. First of all, there's a rise of structural, computational biology tools, AlphaFold, RFDiffusion, BindCraft, bolts, the rise of multi-agent orchestrators like BIOS, Cadence, Lightfold, automated labs like Adaptive, Ginkgo, Chemify that are both built for humans and agents. And right now what we have seen the craze of the OpenClaw, where basically AI is moving from a chatbot to reasoning to fully being agentic and able to orchestrate all of these tools in a sequence. And we believe that biology is becoming fully digitized, agentic science is coming, intelligence is commoditized, and the bottleneck is moved to the wet lab. So what is Peptide? Peptide is, in simple terms, actually not so simple, but I'm trying my best, agentic fleet of swarms that designs, validates, pays for, and learns from peptide experiments, but using blockchain rails. So we close effectively the loop in between computational design and experimental biology, without the person in the middle of the transaction. We use the X402 protocol developed by Coinbase to be able to pay for the experiments really quickly. It designs the candidates, scores them, filters them, pays to lab. In this case, the first one is adaptive, and reads the results back. So we choose the targets, the standards, and it does the work in between. We're really trying to build a connective tissue in between what is happening in silico and what's happening in the lab. So the hard part is not really the model anymore. Intelligence is commoditized nowadays. Everything is just a prediction until it goes to the lab. The only measurement of success is binding affinity in the case of the first gate that we operate after our computational predictions. And basically, peptide really works in the wiring in between prediction and the experiment, choosing the right assay, where do we place the tag, how do we synthesize, and all the orchestration in between. We're also incorporating the new coin-to-company model that Ben this morning explained. So we launched the peptide token using an ignition cell on Bio. We have the treasury that was filled using the USDC stablecoin, and it seeded the first round of experiments. All the research runs live, so the agent is able to design, validate, and pay for the experiments 24/7. You'll find a QR code where you're able to see our website and watch the agent think. Everything is also using on-chain IP, so kudos to the Molecule Labs teams, because now the candidates can be all minted as IP-NFTs in Molecule, so there's full provenance and ownership. That is also a big problem in bigger size biotech companies and pharma. And we're recently moving towards having Peptide LLC, so it reconciles the digital assets with real-world corporate equity and allowing token holders to legally share in the ownership and success of scientific research. A little bit on the architecture of how this is constructed. So the full stack is really on-chain, end-to-end. First of all, the agent framework that we use, you can bring your own harness, you can use Hermes, OpenClaw. We use an instance per receptor program. Each agent has its own memory, history, tools, and basically it orchestrates everything from the design all the way to the lab and reading the results back. The funding and governance is used bioprotocol, so our token is live since 2 months on Base. There's a multi-sig treasury where it settles in USDC, and we also have an Open Labs collaboration. There was Ryan also talking about Open Labs, where we're basically helping SpineDAO design a peptide, and we're doing it fully open so that you're able to see the just how the iteration goes. And everything is, as I mentioned, published on Molecule Labs, on-chain with a timestamp, on hash, and we can mint for interesting libraries, IP-NFTs, and show on-chain provenance. More on the harness. So the harness has 6 capabilities, basically. The heartbeat, model choice, we can choose any of the things that you want to do. For instance, Julian mentioned Grok and GPT were the GPT 5.2, now 5.5 were the best ones, so you're able to choose the model of your choice. They all have their own skills. For example, you want to send the experiment and it's able to install the skill. They have cron jobs that are repeated loops. Channels, we use Telegram or Discord to be able to steer the agent. And the agent have their own memory, how they learn, and they have also a shared memory. We're working on this right now with Martin. A little bit on the pipeline that is running at the moment. We have a bunch of GPCRs, G-coupled protein receptors, like the KISS1R, which is involved in fertility and hypogonadism, OX2R, that is involved in the sleep and wakefulness cycle, and ADHD. Of course, we're targeting the GLP-1R receptor as a benchmark involved in metabolic health. VEGFR2, that it helped us close the loop and show that we're able to design and get results back in a closed loop with adaptive. It's involved in oncology. We also have a program, experimental program running on BPC-157 to design other analogs or work with delivery systems. And we're scoping a new round of receptors. So how it works is that after the computational work that we do, we go to synthesis, either natural amino acids that are very easy to synthesize using cell-free protein synthesis, for example, and we also incorporate non-canonical or non-natural amino acids that are a little bit more complicated and require specialized synthesis and vendors. Binding assay using surface plasmon resonance with adaptive, for example, to be able to provide binding data and kinetics. Then the later stages is cell functional assays for the GPCR signaling. So does it actually switch the receptor or does it have off-target effects? And later stages would be, for example, pharmacokinetics, pharmacodynamics in animal models. A little bit more in detail on these programs. First, we have the KISS1R, as I mentioned, involved in fertility, and basically restoring kispeptin signaling could replace pulsatile GnRH pumps, and it's a disorder that affects 1 in 4,000, 10,000. It's a rare disease as well. And there's 2 leads especially that we designed with one of our partners called LightFold. They all incorporate non-canonical amino acids, and at the moment we've scoped all the assays, all the CROs, and orchestrated this whole operation. So we haven't signed off anything yet, but we can tomorrow if we want it. We also have the OX2R program, which orexin hypofunction is linked to ADHD and narcolepsy. All the approved drugs are antagonists, but we're designing an agonist. All are small molecules, and we wanted to design a peptide. All the rest of the information that I will provide is computationally, but we also have scoped the assays with CROs to be able to prove it in vitro. So we design a stabilized variant that is able to lift the proteolytic activity from 5 to 55 minutes, and this is one of the things that peptides have, that it's the problem with sometimes that just the half-life is very short. So we're working on that. Then we have the GLP-1R. We're not trying to beat semaglutide or tirzepatide, but because there's so much literature here, it helps us ground our models and just the fitness functions in literature review as a to provide as a benchmark. Interestingly, Adaptive also provides assays on GLP-1R that need to be biotinated, so it's a vehicle for us to show also the autonomous loop. And then we have the VEGFR2, which is the one that we sent to Adaptive, and thanks to their fast turnaround, we got results last night. And we used RFDiffusion 3 and ligand MPNN. So we have already agent got some learnings as to what is the next round for design. So what does it look like after binding is one of the things that we discussed before. We want to also map out the full space so that how we can go from sequence, let's say, to dose response. We have already scoped assays and partners, for instance, for synthesis of non-natural amino acids. Functional assays, we use calcium flux assay for the KIS1R and OX2R that are type A GPCRs, and they work similarly in how we assay them in the cell. The readout, it's a 10-point dose response, and as you can see on the right is how it would look like. The references, for anyone that is interested, is called Kispeptin 10 and Orexin B. And we also check for selectivity, whether there's any off-target effects. So taking a step back, where it all connects is basically that design meets measurement. We go from the digital model to the wet lab, we send the candidates and we get the results back. And our goal is to collapse those walls and go to very quick, rapid iteration cycles from designing, building, testing, and learning. So when the agent finishes the computational work, it can place the order through the API, for instance, with Adaptive or other CROs, and it can settle the build through USDC using the X402 protocol, so crypto rails, and that shapes the next round of analysis when the results came back. The first run that we did to effectively show that we can close the loop was, very quickly. So VEGFR2, and it was deliberately chosen to be a flat, hard interface. We designed thousands of candidates using RFDiffusion 3. We also designed some mutants. The positive control shows positive binding, and then a mutant also shows positive binding. No binding for the negative control, so assay is working, and for the other rational mutant that we designed. So that also shapes how we design the next round of analysis. So next one is we chose a target deliberately hard, which is flat polar interface. For the next round, we might aim at another binding site, and we will switch the generator from RFDiffusion BindCraft to BindCraft and avoid glycine and asparagine hotspots. So just to close the loop, closing the loop is a demo in this case, and owning really the data is at each turn is the mode. And as I mentioned before, foundational models are commoditizing, hyperscalers have entered the system, and any well-funded platform can do all of this, but the accumulated set of paired sequence result measurements is really what gives the value, and everything is, all the provenance is attested on chain, so really the mode is the data. So really quickly, what I've learned to working with the agent, sorry Ella, I promise. So the wet lab is the only real gate, the rest is just a prediction. You have to calibrate before you rank with known biology. A failure is a finding, so it's all training data, no waste. I make it check every time, 2, 3, 4 times to make sure that it's working correctly. And what you send to the bench or to the lab really shapes, so we try to spread different designs more than the safe copies of the same one. And while we wait for the results, while the lab runs, we read new papers, ingest it to the agent, more designs, more diversity. Finally, I want to close with this. Coefficient Bio was acquired at the beginning of this year, I think, by Anthropic. Nathan Seafray, the CEO of Coefficient Bio, now is leading Anthropic's life sciences arm. And basically this tweet, I think it reflects where we try to move, where it's a pipeline in a person. It's when one skilled biotech operator with a bunch of agents is able to run a virtual biotech company. So we choose the target, which receptor, which disease, which site. We set the standards, we read the big results, we sign off on the expenses, but the rest is done by the agent. So PepAI designs the molecule, pays for the test, learns from the results out in the open. Thank you, and thank you also to our partners, Molecule, Bio, Lightfall, and Adaptive. And you can check our website here to see how the agent thinks on the latest results. Thank you.